Hey everyone, and welcome back for another deep dive. Today we're going to be looking at how all that data from before human testing, like toxicology, pharmacokinetics, and how well a drug works, all get pulled together for that big step of applying for an IND application.
Yeah, it's kind of like putting together a puzzle, making sure all the pieces fit to tell the FDA the complete story of the drug.
Exactly. And to really understand this, we're going to start by doing a deep dive into a textbook chapter on, get this, basic pharmacokinetics.
You might be wondering, why are we going back to the basics? But understanding these core principles will honestly make the whole IND process much clearer.
Trust me on this one. Plus, we'll uncover some cool facts along the way. Did you know that a drug can actually seem to spread out in a space larger than your body?
It's true. It's called the apparent volume of distribution.
We'll explain how that's even possible a little bit later.
What I think is so cool is that all this basic knowledge is what helps us understand how a drug will behave in a living organism, which is exactly what the FDA needs to know before they approve an IND.
Okay, let's back up for a second. First things first. What exactly is an IND?
An IND is basically a request to the FDA asking for permission to test a new drug in humans. It's the gatekeeper to doing clinical trials. And without it, well, you can't test on humans.
So it's like a permission slip, but with a ton of emphasis on safety and showing that the potential benefits are greater than the risks.
That's a great way to put it.
So then what kind of information goes into this super important permission slip?
Well, a ton of data goes into it. It's not just a collection of random facts and figures either. The IND has to tell a really clear and compelling story about what this drug does in a living being.
Okay, so let's start with toxicology. Before we even think about giving a drug to a person, we need to know for sure it's not going to hurt them, right?
Exactly. That's where animal studies are super important. They let us figure out how toxic a drug is.
So we're talking about like those lab rats, those classic experiments.
Yeah. Yep, pretty much. But there are really important ethical considerations. Scientists always try to gain as much information as possible from the smallest number of animals.
That makes sense. So you're not just trying to find out if a drug is flat out dangerous, but also if it causes any side effects, right?
Yes. We give the animals different doses of the drug, sometimes for different lengths of time, to see how they respond. The goal is to figure out how safe a drug is so we can give people the right dose in a clinical trial.
Okay, so toxicology helps us understand the risks. But what about how well the drug works? Does that matter at this stage?
Oh yeah, definitely. The IND needs to show that the drug can actually treat whatever condition it's designed for. That's where efficacy studies come in.
So you're saying it's not enough to just show the drug doesn't harm the animals. You also have to show it actually does something.
Exactly. The FDA is not going to approve human trials unless there's a good reason to think the drug might actually help people.
Okay, I got it. Now let's get into pharmacokinetics. I think this is where things start to get kind of complicated.
Oh, it's not that bad. Imagine pouring some dye into a container that has all these different compartments representing the different organs in the body. So pharmacokinetics is basically the study of how that dye spreads, how fast it goes into each compartment, how long it stays there, and then eventually how it gets eliminated.
Okay, so basically it's all about how the drug moves through the body.
You got it. And PK data is really important for the IND because it helps us to determine the right dose of the drug to give to people and also how often they need to take it.
Makes sense. Giving too much of a drug or not enough or maybe at the wrong time could be ineffective or even dangerous.
That's right. We need to know how much of the drug makes it into the blood, how long it stays there, and how it eventually gets broken down and leaves the body.
And scientists figure this all out by measuring how much drug is in the animal's blood after they give it to them.
Yeah, they take measurements at different points in time to get a picture of the drug's journey through the body, how well it's absorbed, distributed, metabolized, and eliminated.
Okay, this is starting to become more clear. But remember that thing we talked about before? The apparent volume of distribution, the one that's sometimes bigger than the body. Can we explain that a little more?
Sure, it may sound weird, but the apparent volume of distribution, or VIDA, isn't actually a real volume. It's a calculated value that tells us how much of the drug is in the tissues compared to the blood.
So going back to that dye analogy, it's like some of the dye is sticking to the sides of the container, making it seem like there's less dye in the liquid.
You got it. That's a perfect analogy. If a drug likes to bind to tissues, it will look like it's disappearing from the blood, and that gives it a larger VID.
So large V dye doesn't mean the drug is literally taking up more space than the body. It just means it's more spread out in the tissues.
Exactly. And here's where things get really interesting. Our source material gives some specific examples. Drugs that stay mainly in the blood, like heparin, have a VEED that's about the same as the blood volume. But drugs that prefer fatty tissues, like amphetamine, can have a VEED of up to 200 liters.
Wow, 200 liters, that's a lot. But why should we care about the VEED? Does this really matter in the real world?
Yeah, for sure. VEED helps us to predict how much drug actually reaches its target in the body. So a drug with a large VEED might need a higher dose to have the same effect as a drug with a small VEED.
So it's all about getting the right amount of drug to the right place in the body. And that's why PK data is so important for figuring out safe and effective doses.
Exactly. VEED is just one piece of the puzzle, though. We also have to think about things like elimination half-life, which is how long it takes for the body to get rid of half of the drug.
Right, that makes sense. If you know how long a drug stays in the body, you can figure out how often someone needs to take it.
Exactly. Elimination half-life can be really different depending on the drug. And it can also be affected by things like how well someone's liver or kidneys are working.
So if a drug doesn't stay in the body very long, someone might need to take it multiple times a day to keep enough of it in their system, right? But if it sticks around for a while, they might only need to take it once a day or even less often.
You're getting it. Understanding a drug's half-life is really important for coming up with a dosing schedule that makes sure it works the way it should without causing any problems.
Okay, I think we've covered a lot of ground here. We've talked about how important the IND is, the different kinds of studies that go into it, and even the basics of pharmacokinetics, including that crazy concept of apparent volume of distribution.
We've done a lot, but there's still so much more to talk about.
There is.
So stay tuned for part 2 of our deep dive, where we'll get even deeper into the details of IND applications. Welcome back for part 2 of our deep dive into IND applications. Last time, we left off talking about elimination half-life and how important it is for figuring out dosing schedules.
Right, right. But this time, let's focus on another big piece of the IND,
Right.
the efficacy data.
Okay.
It's great if a drug doesn't hurt the animals in preclinical testing, but does it actually work? That's the big question, right?
You're right. We need to know for sure that this new drug can treat the condition it's designed for. And that's where efficacy studies come in.
So these studies, they basically involve giving the drug to animals that have the disease you're targeting, right? Like if you were developing a drug for high blood pressure, you'd give it to animals with hypertension and see if it lowers their blood pressure.
Exactly. That's the basic idea. And there are a bunch of different ways to measure efficacy depending on what kind of drug you're studying. Like, if it's a new antibiotic, you might infect animals with the specific bacteria and see if the drug kills the bacteria or at least stops it from growing.
Gotcha. So efficacy data is all about showing that the drug can actually do what it's supposed to do. It's not just about safety.
Right, and this data is a really important part of the IND application. It shows the FDA that there's a good chance the drug will work in humans, not just that it won't cause any immediate harm.
Okay, that makes sense. So now we have toxicology data to figure out the risks, efficacy data to show the potential benefits, and PK to help us figure out dosing. It seems like that's a lot of information to put together for the FDA, though.
Oh, it definitely is. And that's where the challenge of the IND comes in. You're basically putting together all this preclinical data to make a convincing argument for why this drug should be tested in people.
So it's not just like throwing a bunch of data at the FDA and hoping for the best.
No, not at all. The IND has to tell a story. And that story needs to make sense to the people who are reviewing it.
Okay, so it's got to be organized, right?
Yes, exactly. For starters, it has to be organized in a very specific way. It needs clear labeling, indexing, all that good stuff. The FDA reviewers need to be able to find the information they need quickly and easily. They have tons of these applications to review, so being clear and organized is super important.
Yeah, that makes sense. But it's more than just organization, right? I mean, there are rules about what kinds of studies need to be done, the statistics that need to be used, and how the results should be presented.
Right. You're exactly right. There are these very specific guidelines that come from both the FDA and international organizations like the ICH, the International Council for Harmonization. And these guidelines, they set the standards for all of this.
The ICH, what is that exactly? Some kind of international drug development police force?
Well, not exactly a police force,
Yeah.
but they are pretty important. The ICH is a group of regulatory authorities and pharma industry experts from all over the world. And they get together to develop these harmonized guidelines for drug development.
So they're kind of like the United Nations of drug development.
Yeah, something like that. They're trying to make sure that new drugs are developed in a way that meets high standards for quality, safety, and efficacy everywhere.
Okay, so the data is organized and is following all the rules and guidelines. What else needs to happen before you can hit that submit button?
Well, remember how we talked about the IND telling a story? That story needs to be, well, a good one. It needs to answer all the questions that the FDA reviewers will have.
Like, is this drug even worth testing in humans?
Exactly. The IND has to make a strong argument based on solid scientific evidence that the drug has the potential to benefit people.
And I'm guessing it also needs to address any potential safety concerns.
Of course. It has to outline a plan for managing and monitoring any risks that might come up during clinical trials. It has to be very clear that patient safety is the top priority.
So being honest about what you don't know and having a plan to deal with it if something unexpected happens.
Yeah, pretty much. And all of this information, it goes into a document that can be, honestly, hundreds of pages long.
Hundreds of pages. Wow.
Yeah.
It really shows how much work goes into developing a new drug.
It's a huge undertaking, but it's an important one. It's all about making sure that new drugs are tested thoroughly before they're given to people.
Okay, so we've talked about how the IND is structured, the guidelines it has to follow, and the fact that it needs to tell a convincing story. What else do we need to know about putting it all together?
Well, one really important thing is how all the different types of data fit together. Remember, we have data from toxicology studies, efficacy studies, and PK studies. All of these pieces need to work together to tell the complete story of the drug.
So it's like putting together a puzzle.
Exactly. A really complicated puzzle. And this is where the people who put together the IND really need to be experts. They need to be able to see the big picture, spot any problems with the data, and then draw conclusions from all of this information.
Sounds like it takes a lot of skill.
It definitely does. It's not just about summarizing what each study found. It's about figuring out what it all means together.
So each data point is kind of like a character in a story, and the IND team has to figure out how to weave all those individual stories into one big narrative.
That's a great way to think about it. And that narrative has to be clear, easy to understand, and convincing. The FDA reviewers need to be able to follow the logic, agree with the conclusions, and ultimately believe that this drug has the potential to be both safe and effective for people.
So no pressure.
Right. But seriously, it is a big responsibility.
Okay, so can we look at some real-world examples to see how all of this data integration works in practice?
Sure, let's say we're developing a new cancer drug. In the toxicology studies, we see that the drug can cause some liver damage, but only at really high doses. But then in the efficacy studies, we see that the drug is super effective at shrinking tumors.
So now you have a tough decision to make.
Yeah.
Do you risk the potential liver damage to get the benefits of shrinking tumors?
Right. It's a classic risk-benefit situation. And that's where data integration is so important. We can look at the PK data to see if we can get the same tumor shrinking effects at a lower dose, a dose that's less likely to cause liver damage.
So you're trying to find the sweet spot, the dose that gives you the most benefit with the least amount of risk.
Exactly. Now let's imagine that the PK data shows that the drug is eliminated from the body slowly. That could mean that patients wouldn't need to take it as often, which might also help reduce the risk of side effects.
I'm starting to see how all these pieces fit together. But what about those regulatory guidelines we were talking about before? How do they affect the way you integrate the data?
Well, remember that the FDA and the ICH have all these rules about how data should be analyzed and presented. So when we're putting together the toxicology, efficacy, and PK data, we need to make sure we're following those rules very carefully.
So you're not just drawing your own conclusions, you're also making sure you present those conclusions in a way that meets the FDA's expectations.
Right. If the data isn't presented the right way, the FDA could reject the whole application.
Wow, that's pretty intense.
Yeah.
Seems like developing a new drug is not only about doing good science, but also about being really good at communicating that science.
Yeah, that's a really good point. You have to be able to speak the FDA's language, basically.
So in a way, it's like a partnership, right?
It is.
The IND team and the FDA are working together to make sure that new drugs are safe and effective.
Yes, that's exactly what it is. A partnership.
And we're back for the final part of our deep dive into IND applications.
We've come a long way. From the very beginning, talking about preclinical research with all that data, from animal studies to where we are now, ready to talk about clinical trials with real people.
Exactly. And all the things we talked about before, toxicology, efficacy studies, all that PK stuff, it's all led up to this moment. This is the chance to see if this new drug really can help people.
Yeah, it's exciting, but There are some challenges too. I mean, animals aren't humans, right?
Of course not.
Yeah.
But how does that difference actually affect how we use preclinical data to design those first human trials?
Well, animal models give us really useful information, but we have to remember that a drug might act differently in different species.
Mhm.
A drug that works great in a mouse, it might not work exactly the same way in a human.
So how do we make that leap? How do we take what we've learned from animals and use it to design safe and effective trials for people?
Well, for starters, the IND has to be upfront about those differences. It needs to be really transparent about the limits of the preclinical data and show exactly how it's going to address those uncertainties when it moves into clinical trials.
So it's like you're admitting you don't know everything and you have a plan for what to do if things don't go as expected.
Yeah, exactly. One of the tools we use to go from animal data to human dosing is allometric scaling.
Allometric scaling.
Yeah, it sounds complicated, but it's basically using math to predict how a drug's behavior will change based on body size.
Okay, so if we know how a drug acts in a tiny mouse, we can use that info to figure out how it might act in a much bigger human.
That's the idea. It's not perfect, but it gives us a good starting point for those early trials. It helps us to figure out a safe starting dose for people.
That makes sense. You don't want to just guess when it comes to giving people new drugs.
Definitely not. And when we're talking about these early trials, we have to consider who those first participants will be. We can't just give the drug to anyone. Choosing the right people for the trial is super important.
Oh, yeah, you need people who represent the group the drug is for and who don't have any other health conditions that could mess up the results, right?
Exactly. The IND has to lay out very specific criteria for who can and can't be in the trial. It's a balancing act, you know?
Yeah.
You want the participants to reflect the real world, but you also need to make sure you get clear data from the trial.
Of course, keeping people safe is the most important thing during the whole process.
Absolutely. We need to be ready for anything that might happen.
So, like, having backup plans in place, just in case.
Yeah, exactly. The IND has to include a detailed plan for how to monitor and manage any side effects, including what to do if anything unexpected happens.
Okay, I got it.
And this leads us to one of the most interesting parts of the IND, how all that preclinical data helps us design the clinical trials.
Right, because all that work we talked about before, it's not just for the animals.
No, definitely not. All that preclinical data is meant to help us design those human trials.
It tells us how much drug to give, who should get the drug, and what to look out for in terms of safety, right?
Yes, and more. The IND is basically a bridge between the animal studies and the human trials.
So you're not just showing the data, you're also explaining why it matters and why it makes sense to move forward with testing the drug in people.
Precisely. And that argument has to be really, really strong. The FDA is going to look at every detail, trying to find any holes in your reasoning.
It's almost like you're presenting a case in court. You have to anticipate what the other side will say and make sure you have all the answers.
That's a great way to think about it. And this back and forth between the IND team and the FDA is really important. It makes sure that the trials are designed in a way that makes sense scientifically and is ethical too.
So it's a real collaboration.
It is.
Both sides are working towards the same goal, which is getting safe and effective new treatments to people.
Exactly. Well, we've talked about a lot in this deep dive into IND applications. We started with preclinical research, then got into all the complexities of data integration, and finally made it to the transition to clinical trials.
It's been quite a journey. I feel like I have a much better understanding now of how new drugs are developed.
And it's just the beginning. The IND is a critical step, and it holds the potential to help a lot of people.
It's been so interesting to see how scientific evidence is used to create real solutions for patients.
We hope this deep dive has helped you to understand the dedication, the expertise, and all the hard work that goes into making new medications.
Thanks for joining us on the deep dive. And keep being curious about the world of science and medicine.